Hello, Assalamu alaikum. In this video, we will understand about the special anti-aging compound, the glucoraphanin, which that convert into the sulforaphane whenever you will intake. So this is the chemical formula which that show in the diagram. So whenever you will intake, so our digestive system contain microbiota, which that produce the enzyme myrosinase, which that convert into the sulforaphane from the glucoraphanin. And this sulforaphane is basically important improvement of high fat gut bacterial flora, which that high fat diet will digest easily. On the other hand, the white adipose tissue convert into the BG adipose tissue and due to the production of more UCP1, and the UCP1 is very important for production of heat. In this way, in the cold weather, that is important. So insulin resistance also decrease and inflammation decrease due to the macrophage 1 activation inhibition, while the macrophage 2 polarization will lead to even inflammation decrease. On the other hand, important thing you should need to understand here is that dysbiosis will decrease in this way. The sulforaphane is very important regulator of the microbiota in our gut, and in this way the dysbiosis prevention. On the other hand, the insulin resistance and inflammation decrease due to the M1 macrophage 1 inhibition to activation. So here is the before the sulforaphane intake, the patient, for example, which that high blood glucose, and in this way the glucose production more, gluconeogenesis increase, insulin resistance increase, oxidative stress and inflammation is more, while the insulin secretion, insulin sensitivity, glucose uptake, and the glucose tolerance and the hepatic glycogen, which that is basically decrease. It means the liver will not store the glycogen, not store the glucose into the glycogen, while the glucose uptake will be decreased. In this way, the problem is that the adipose side will not release the glucose to the liver for deposition that in the form of glycogen, and the pancreas also not working. When you will intake the sulforaphane, that glucose homeostasis will occur. In this, the blood glucose level ultimately manage, and the glucose production decrease while the gluconeogenesis decrease, insulin resistance, oxidative stress, and inflammation will decrease. Because the sulforaphane also activates a certain type of gene for the antioxidation process, for reactive oxygen species prevention, and also insulin secretion increase, insulin sensitivity, glucose uptake, glucose tolerance, and the hepatic glycogen will increase. So that is the beneficial. Let's understand about the further. The basically insulin sensitivity whenever increase due to the sulforaphane intake. In this way, the glucose metabolism increase to produce more ATP. In this way, our body will feel more energized. So let's understand about what happening after the sulforaphane to activate the certain type of the gene and protein. So here is the nuclear receptor F2, which that is increased due to the gene activation lead to antioxidant genes activation, and the P genus kinase and BAX BCL2 will also decrease, will lead to cell death decrease. So in this way, the cell homeostasis and cell stability will increase, while the necrotic factor kappa B for the immune activation will decrease. In this way, the immune system regulation, while hepatic toll-like receptor expression also decrease, and the AMPK pathway will increase for the DNA repair and the damage repair. Modulates the GSK3 beta and FIN and NRF2 increase signaling pathway, and endothelial dysfunction will be improved. In this way, the decrease the endothelial dysfunction for the prevention of the cardiovascular disease. The glucose whenever intake, in this way the insulin sensitivity will increase. But on the other hand, the sulforaphane is very important for the DNA damage prevention. Whenever the UV radiation will damage the DNA due to the reactive oxygen species and free radical, So in this way, the sulforaphane will activate the NRF2, will lead to activate the antioxidant mode of action, will prevent the DNA damage, and in this way the cancer prevention ultimately.